Vale, doncs comencem. Moltes gràcies per venir un altre dia. Today we'll have a seminar by Margarida Barceló, which is a postdoctoral researcher in the ecology lab. And she's a Marie Curie, has a Marie Curie fellowship. And when she will talk about her analysis on the she's expert in the genetics of animal behavior. Now she's applying this to the fish. So, Laura, thank you. The floor is yours.
Thank you, Nico, for the introduction. So, yes, I've been always fascinated by the field of animal behavior, and I also like genetics. So I'm trying to combine both fields. I'm like the middle of two different worlds, but I'm just trying to combine them together. But I think it's multidisciplinarity is what brings best in science. So the field of animal behavior has been studied for centuries now, and is defined as the way that animals interact with their environments, or the way they have to respond to the environment, and also how they interact with other individuals. And traditionally, so there has been these big classifications of different animal behaviors in the way to explain a function. So we have, for example, parental care, that would be different behaviors that try to provide the best for the offspring to ensure their survival. There are other fields that can explore, for example, activity patterns, so differences in between individuals being awake or asleep in response to the changes of the solar cycle, and also, again, like social interactions that can provide with different bonds between individuals to ensure maybe mating, or ensure pair bonds or individual bonds to ensure survival. And over the years, these animal behavioral focus, or some researchers working in animal behavior, they start to look at also the individual level. So differences in between individuals that found differences in behavior that were more, so differences in between individuals that to solve the same problem. And that evolved into the field of animal personality. So in humans, personality is something that is easy to understand. So we know what is a human personality. In animals, sometimes it's a little bit more tricky, but it has been just defined as the consistency of behavior over time and in different situations. So you can see here, for example, this stickleback that it's being presented by a model, and how it shows aggression towards this model. And we will expect that if we do this in different times and maybe in different contexts, this individual is going to show similar levels of aggression, or at least being more aggressive than another individual that was less aggressive than him. And like this, there are many ways that we can classify this animal personality. For now, to quantify this behavior, there are what I call personality axes, like here, like aggression or activity levels, or exploratory behavior of novel objects, like this pen that is presented here to the bird, or exploring new environments. And I'm not going to talk that much to this, because this is the focus of some chapters of Martina's thesis. So hopefully one day she will come and explain all of this animal personality world. But what is interesting to me is this consistency of behavior that I was talking about. So individuals it appears that are not free to display any array of behaviors. They are limited to a specific behaviors that perform what is their personality. And this limitation or this consistency is because these behaviors have a genetic basis. So the genetics of these individuals are making them be limited to the behavioral response they can provide. And that's what I'm studying. So at the beginning, they start looking at these behavioral differences, so the genetics of these behavioral differences at a sequence level. So here we have a chromosome with the DNA. So this DNA is just a chain of chemical structures called nucleotides, that the sequence of these chemical structures, one after the other, have the information to create a protein, okay? And these specific sets of regions of DNA that create its protein is what I call genes. And these genes are responsible to a function. So, and it happens in morphology as well as it happens with behavior. So differences in the sequence of these specific genes are responsible of differences in behavior. For example, we have examples of different genes. So we have the clock gene that has been related in zebrafish with differences in activity patterns, and in zebrafish and also in other species. So even in vertebrates, they have this gene that has been related. Differences in the sequence of this gene have been related into behavioral differences. We also have the dopamine receptor B4 that has been related with exploratory behavior in birds, and the arginine vasopressin receptor 1A that has been related to the formation of pair bonds in these balls. And of course, just talking about differences in sequence, explaining this complex behavior, it's somewhat of a simplistic view of the whole process, but that was the only way we have at the beginning when the technology is only allowed to look at the gene sequence. But now this field evolved a little bit more, and now we are not only looking at one gene, but a large network of genes and how they interact with each other and the expression levels. So we can look at, for example, gene expression, and here is how we can study it. Of course, the tissue or the organ responsible of behavior will be the brain. So we have to look into the brain, so the differences in gene expression in the brain. And we can look here, for example, we have an example where we have behavioral A that will be represented in this column here, and behavioral B, and all of these will be genes. So when an animal is displaying this behavioral A, all of these genes up in this part of the column are being expressed, and these ones are not. Oh, and they are not in behavioral B. And here it will be the opposite. So here we can actually associate a specific genes being turned on or turned off in function of the behavior that this individual is displaying. And this has been found in, for example, honeybees. So when they are honeybees, they have these, they can be foragers, so the ones that go out and look for nectar, or nurses, that the ones that stay in the hive and take care of other individuals in the hive. So depending on if they are doing one function or the other, specific genes on their brains are expressed. The same has been found in sticklebats. So this fish here, individuals displaying being really aggressive or less aggressive, they have different genes turned on and turned off on their brain. And also pair bond and monogamy has also been related in different organisms, not only birds, but also fish and frogs, and different mating strategies have been related to similar genes in different taxa. And you will wonder, and why is this important? So as I said at the beginning, so the animal behavior is the response to the environment, the way that animals have to respond to the environment. And with the changing world we have right now, with climate change and all of these anthropogenic challenges that we are taking to these animals, it is important to know the mechanism, how these mechanisms work in order, if we have to create management techniques or things like that, to be able to ensure biodiversity maintenance. So I'm going to take this a little bit closer to home. So as Aneko said, I'm working at the Fish Ecology Lab, and we work in we are interested in the study how fisheries affect fish populations. So one trait one concept that I want you to understand, that is a little bit, it's not related with behavior, but it's something that you might be familiar with. It's the trait selective harvesting. So in fisheries, it is known that there is a preference for bigger individuals. So either by law or by commercial preferences, in a fishery, we always catch the individuals that are larger. So after many, many years of selecting this specific type of individuals, the populations of fishes have shifted their sizes, and now individuals are smaller. And also the reproductive output is also smaller because females don't grow as much, so they cannot produce that many eggs. So we can see how this trait selective harvesting is having an effect on fish populations. And we were wondering what is happening with behavior. So we have seen that there are certain behaviors that could be affecting the way that animals are harvested. So for example, imagine an individual that is more exploratory. It could be that this tendency to explore is going to make it fall more easily into a fishing net, for example. So we could be potentially harvesting preferentially these individuals. In the same way, individuals that are more active or that start their activity earlier, they can also be easier to catch, or at least they are going to be exposed for a longer time to fishing pressure. So it is possible that these individuals are going to be differentially harvested. So over many years of exploration, these behavioral traits are going to be selected again and can be removed from the population. This, of course, it will have an effect on the genetic variability of the population. So in order to study how all of these behavior and genetics are connected into the fisheries world, we are studying the pearly razorfish as one of the species. Probably some of you already know. We are all the time doing things with the little fish. This fish is a labrid that inhabits coastal sandy bottoms of the Mediterranean and also the Atlantic in both temperate waters of both coasts. It's a protogenic hermaphrodite. It means that it's born as a female, and when it reaches a certain size, it becomes male. It performs this characteristic burrowing behavior when individuals burrow in the sand at night for protection and rest. And that's actually really good because it allows us to measure the onset of activity of these individuals, so we know at what time they start their activity in the morning. They also form family groups formed with one male and a few females, and they are heavily territorial, so they defend the territory, so we can get also a measure of aggression on these species. And they are also heavily harvested by recreational fisheries. So during the reproductive season, there is a closed season, so it cannot be fished. And on September 1st, they open the season, and all the fishermen, they go out and fish them like crazy. And we have calculated that in the heavily harvested populations, 60% of the population is removed in only 2 weeks. So it gives us a really good experimental setup where we can study how this fishing pressure is affecting either the behavior and the genetics of the species. So when I started working with these species, there was no genetic information available. There was no studies done. So we had to start from the beginning, from the basis. And we start looking at this clock gene that I was talking at the beginning. So the gene that is related with sleep and activity patterns. And I focused the research on a region that has been found in other species to be related with this activity rest pattern. That is a region on the terminal region of the protein that is called the Q-rich region. It's just an area that there are many amino acid repeats. So the same amino acid is repeated multiple times. So each line represents one individual. So as you can see, there is a little bit of variability. We have 4 different versions of this gene that are found naturally in our population. And I compared the different versions of this gene that one individual has to the activity patterns of this individual. And I found that individuals that started their activity earlier in the morning had more repeats of this amino acid on their genetic sequence. So we can find here an association between the genetics and the activity patterns, leading to a possible trait selective harvesting in this case. And I'm also right now working on this DRB4, so the other gene that was related with exploratory behavior. So we have exploratory behaviors, we have exploratory measures for these species, and I wanted to check if I was able to find also this variability in the same area of the gene that was described for this bird species. So here again, we have the genetic sequence is each one of the lines, so each individual represents one line. The areas where you find the same color are areas that are not the same, but areas like here that I can zoom in this area, there is it's called the SNP, so it's just like a single nucleotide polymorphism that is variable between individuals, and we can compare this variability at the genetic level and compare it to the variability at the behavioral level. I don't have the results of this yet, sorry, but I hope I will have this soon. And of course, that was the beginning, but now we decided that we needed to go a little bit further and start exploring what we were talking about before and look at the gene expression of the brain. So look at if there are gene expression changes at the brain level. And that's the preliminary results we have from this analysis. And this Each point of this graph should represent a gene. It doesn't, because it's a little bit fragmented, so we are still working on that. But any gene that is above this line or on either side of these two lines will be genes that are differentially expressed. So in between individuals that wake up early or individuals that wake up late. So it means that we can find a network of genes that are related to the wake up hour preference. As I said, this is just preliminary because we haven't been able to obtain complete genes. We just have fragments. That's why we put that project on hold, and we got funding to create the reference genome for the species. This is just to get the entire gene sequence for all of the chromosomes of all of the for the entire genome of the individual. And with this, we can use this reference genome to be able to have the complete genes on this gene expression analysis, and then we'll be able to get some results from that. And once we are done with all of this, we will be able to have a better idea on what genes are related to what behaviors. And this way we'll be able to have a better idea if there is the potential for this trait selective harvesting that I was talking before. So, as I said, there could be that if we are selecting out these specific behaviors, we are also selecting out these specific genes. So we could be in a scenario where in marine protected areas, we find all of the genetic variability because these fish are not being captured. So we can find all of the genetic variants, all of the diversity that I was talking before. And in heavily harvested places, it could be that all of these species that were easy to capture, they are removed from populations. Actually, we can see how this could lead to a place where we still have fishes on the population, but are the ones that they are really hard to fish because the ones that they were easy, they have been already removed. So fishermen, they can come and say, oh, there are no fish. Like, yes, there is fish, but it's very, very hard to capture because you have selected out the ones that were easy to get. So with this, we are also, so that's the first part of the project. And in the second part of the project, we also want to look at the genetic connectivity between different populations of the Balearic Islands. So this is just a simulation of how the hydrodynamic currents move in between the Balearic Islands. And these species, they release the eggs and larvae to the water column. So we can imagine how different populations, they can be releasing the eggs and larvae, and these eggs can disperse in different areas. So we can imagine that if we have a marine protected area in a place that it's heavily dispersed, this marine protected area can serve as a reservoir of genetic diversity that can compensate for the losses that are provoked due to fishing. So, well, that's all I want to tell you. I think I went a little bit fast, but I hope you understood and I can answer any questions. Thank you. Yes.
So, very interesting. I have an array of questions, a few of which I'll ask you later. But okay. So for the association between that you found already, for you have the clock gene, yes, and the activity, you could do. So your idea is that might explain part of the selection that is being done or has been done in the harvested areas.
So well, now the samples that we have are from a marine protected area, so we would expect there to have all of the variety. So to have the ones that start early and the ones that start late, because they should be all there. But
my point was because there isn't well, at least it doesn't look like there is a difference. There isn't a dramatic difference there isn't a dramatic association. There is a bit of association. So what I was wondering is, would it be possible to estimate what sort of selection would this by itself cause with quick mathematical model, for example?
Well, I mean, there are ways to look at how if there are selection pressures against the specific regions of the genome, because that leaves, so there are mathematical ways to calculate the selection pressures. Yeah. So yeah, there are ways. We haven't done it with this specific gene, but we plan to do it, but more on a whole genome scale.
But in that case, you would only see an association. You wouldn't see a
yeah, of course. Yes, it's just an association. Yeah, and of course, and many times. So the main problem with this associating one particular gene of one region with one particular population is that normally every time that someone did that, when they repeat the exact same in another population, the association is not there anymore. So that's why they the field change, and now everyone is looking more at the whole transcriptome level, so looking at the genetic network level, because when you look at one specific region, sometimes it works and sometimes it doesn't. So it's not that big of finding, but it's the first thing I have, so I always stand on it. But it's not, yeah. And also it will be interesting to see if we find, so if we look at this specific region on places where there is a lot of fishing, to see if we only find one particular type of.
I didn't mean to.
No, it's fine, it's fine. No.
Well, I don't know much about this, but anyways, I would say that the character of an organism is probably induced, for sure is induced by genetic reasons. But there is, in some percentage, there is also something that is learned from the epigenetics, from the behavior. So, as far as I understood, you try basically to explain all the behavior just by genetic things. So do you think that there is a bias there that actually a part of that population is behaving like this because of, and particularly in a fish where it's got some territorial behavior and group behavior, is actually something that has been learned from other fish, and also if the behavior is something that is associated with age and with sex, because those organisms are basically changing their sex. And over that change, if there is also a change in behavior.
Yeah, well, at least in activity patterns there is. So at least males, they start the activity earlier than females, and we think it's because they are very territorial. We think that they might be starting their activity to ensure the protection of the territory boundaries. But what is interesting for this species is that they have the exact genetic sequence when they are males or females. That's something that it's not, it doesn't happen in mammals or other species where the sex determination comes by the genetics. In this species, it comes by gene expression, but not by gene sequence. So I think it's very interesting to study this and see how the same individual with the same genetic background can behave differently when it's male and when it's female. And that would be something that would be great if we could keep the same individual and test the behavior in both cases. So yeah, that would be, and also I think that the major part of the animal personality literature is done in birds. That is also, they have this chromosomal sex determination. So it would be really cool to see how that plays on a species that has this sex change. So I don't know if I don't think I can answer your question, but it would be great if we could do some experiments and look at that, because for sure, I mean, there are many things that the environment has a huge effect on behavior. But I feel like what is interesting is like, yes, the environment has a great deal on the behavior, but still there is something that is outside, and that's actually the selection pressure acts on the genotype, not on the, because it's the, so what it's passed on to the next generation, at least for these individuals, is only the genes. So the offspring cannot learn from their parents because the eggs are released with water column and lost. So the only thing that is transferred from one generation to the other is the genetics.
I'm going to a higher level. How does this affect to domestication of some species? Because there are, for example, wolves and certain species of dogs were more easily domesticated because they have certain behaviors that are more easily adapted.
Well, I mean,
I know that has nothing to do.
yeah, I'm not that familiar. I know there is an entire field of working on domestication, genetics on underlying the domestication, because there are species that you cannot domesticate, and so probably there is something there too that you can. And of course, there are you can see it in artificial selection experiments that you can, with the same, starting with the same genetic background, you can get like completely different forms, like in dogs and stuff like that. So I don't know that much about this. Yes.
My question is much more you define your personality as the expression of genetic. When you go to more complex individuals, humans, for example, you can say something that the personality is driven by the population of genes.
Yes, there are studies that show that there is one of the genes, I haven't talked about it, but there is one gene that the experiment was done in humans, and it was kind of like, it's a gene that is related with foraging behavior in many species. So the way that individuals forage so they go really far or explore more in the area. So they look at genetic differences that were related to how far you go. And they gave to little children an iPad, and they were playing a game that was a foraging game. They were supposed to go and like tap on different things, and they found also this relationship. So the behavior of the children playing with that iPad was also related to the exploratory behavior found in other species, and it was related to the similar genetic underlying. So there are studies that show that humans have, on a different extent, of course, but there are some genetic markers that can kind of define human personality too.
This is related to what they said. I wonder, you say aggressive behavior or non-aggressive, but probably what the animals that survive the best are those that are able to adapt.
Mm-hmm.
And this probably means that they need to change the behavior depending on the circumstances. So I wonder if there are some genetics on that, on how flexible the behavior is. And
yes, there is. So
whether you look at this flexibility on the behavior instead of.
Yeah, there's always this contraposition of like the behavioral plasticity and genetic determination, and it was always seen like, oh, you are either plastic or you are defined by your genetic makeup. And actually, they have been found genes that are related to this plasticity. So
that was what I was asking, because probably what is making
so it makes you more
survive the best is plasticity.
Yeah.
This is probably heritage as well.
Yes, it is. But the problem is there are some genes that regulate different traits. So sometimes maybe the same gene can be responsible of a trait that is very beneficial for you in one situation, and then when you go into another situation, it's not that good. So it could be that maybe it's not that easy to say, oh, you have to be very plastic, but that sometimes maybe it depends on the context also where you are. So probably there is no best strategy there, but
genetic plasticity or the
the
plasticity that is codified genetically, this is linked to different genes, or is it something more?
Probably, I don't know exactly.
I don't know.
But I would think that it will depend a lot on what behavioral trait you are also talking to, because I mean it's very complex. So I don't think it's like one gene for explaining the behavioral plasticity for any behavior that you can find. So probably it depends on the specific behavior you are talking about. You will have, and there are going to be behaviors that are more plastic than others. So depending on how crucial it is, maybe in your survival, it will have more flexibility.
And have you found species that are more plastic in their behavior than others, and this to be linked to the
we haven't analyzed the much data yet, so we are still working on this. So I don't know, but probably we'll find some.
Very interesting.
Yes.
from a population point of view, you have a very strong directional selection towards larger animals and bold animals. So, do you have any idea of gene preferences? Because also your sample is skewed behavioural. So when you catch a fish, it's more likely to be a large fish than a bold fish. Do you have any idea of that? Because with 60% of the population removed every year, either you have a lot of immigration or you lose your population in 2 years.
Yeah, so we don't know exactly how is it possible that we still have fish in there, but probably, so I would think that there is the immigration from these larvae coming in from other places, and probably maybe, I don't know, maybe from adjacent places that are close by that they can get. It has to be immigration, because if not, we would not have fish anymore. So so yes, and of course we are skewed with, because these fishes are really hard to capture in a different way that it's not by fishing. So that's our main problem, and we are aware of that. We have tried to try to find some other ways, but the problem is, like, when you go, if you try to catch them by scuba diving or something, it's almost impossible, because they will escape. Or even if you try with nets or something, they will burrow in the sand, so you are not going to save them. That's also why they are very expensive probably in the market, because you have to go there with the fish and line. So we are actually taking an obscure representation, because when we do our experiments, we take only the ones that are easy to capture. But I would think that with intense sampling and comparing harvested versus non-harvested populations, we at least will be able to see if there is an effect. Yes.
So when you have protected areas, you have high variability, higher than where you can fish. And you said about the dispersion of the larvae, so that they're feeding these areas that are overfished, and that's why you don't have. So could you, so it's super hard, you just said, but could you see a variability whether you go harvesting, taking fishes, before the fish season is open again, and after? Because if it's the case that you have these areas that are radiating, spreading eggs, and then they go there, if you, in your samples, do you make a difference between the time of?
Yeah, we could do that, like before and after treatment of after the fishermen. Yeah, we could do that. We haven't done that yet, but probably when we start like developing more into this, that's something that we could do. And also, so we are comparing. So every year you have this new input, but the fishes that are older, they are supposed to be there because they survived all of these waves of fishing. So I would assume that the genetic diversity will also be reduced the older the individual you get. So we see there are like different ways to measure. It's really good to mention. Maybe the next seminar in a few years I can tell you more.
Good question. I didn't understand. I didn't understand how you measure gene expression versus just the
sequence.
sequence.
So yeah, there is two different technologies. So the gene sequence, yeah, I didn't talk that much about the methodology, but the gene sequence, you can get it from any cell in the body. So what we normally take is just a small thin clip from the fish, so we don't have to kill it. And from any cell in the body, you can get the chromosomal information and the DNA. So you can just get the sequence from there. But the problem with the gene expression is that you are measuring the products of the gene. And for that, you have to take it from the sample so from the tissue that is producing this, right? So that in this case is the brain. So you need to collect brain samples, and what you do is just extract what is called the messenger RNA. So you have like the sequence of the DNA, then you have a messenger RNA, and then you have the protein. So you sequence this intermediate product, and the abundance of this intermediate product, it gives you a level of expression of this gene.
I can't. If you have a isogenic population or a community that has the same genes, it's clonal. For example, in humans, for example, twins. how often, I'm sure that there must be studies on behavioral differences between twins, right?
Yeah, the natural is
But even without being so drastic, so how, in that case, you have the same genetic background, but it's not clear to me that this genetic background is expressed in the same way in the two twins or more twins. I don't know. So, what is actually, is it the genetics that is more closely related to behavior, or is the expression level that is mostly related to behavior? Because expression, I would suppose, depends also on the environment that you find yourself in.
Yeah, I feel like, see, for these twin studies, I find it fascinating, and I start like reading them because I think it's super cool that you can do this with humans. And they found that actually, no, not the things, but like studies in humans. And I found that there was a study where they were looking at behavioral differences between twins that were that grow up together and twins that grow apart. And the ones that grew up apart were more similar behaviorally than the ones that grew up together. So it means that like growing up together, they were trying to make a difference between each other, because probably they didn't want them to like, but the ones that grew up separate, they actually look more alike. So it appears that of course there is an effect of the environment, but there is also a genetic underlying conditioning the behavior of the twins. But there is also another experiment that it was done in, I think they were fishes, I think they were clones, and they had the exact same environmental conditions, and they found behavioral differences in those two. So it was the same genetics, same environment, and they behaved a little bit different. So that was also very cool, that it shows you that even the environment, there is always some variability added to the system that can turn into behavior variability.
That's actually, how do you define the behavior? Just like agility, or because, I mean, a person is listening, but in a fish, I guess you just do how much it moves, or?
Yeah, it depends on what you want to do. So for example, for personality, that's mainly what we are doing in the lab. So those are standardized tests that you do. For example, with the pearly razorfish, we did what it's called the mirror test to measure aggressiveness. So you put a mirror in front of the fish and you look at how many times it attacks the mirror, and you can count the attacks or count and this time we're counting the attacks and the approaches to the mirror. So it's like kind of like aggressive displays.
And it was very aggressive, it's narcissist.
Well, apparently these species, at least we believe that they don't recognize themselves to the mirror. So it should be that they are just like attacking an opponent. Yeah. And then we also put like a novel object to see how they were exploring. And in this way, we were looking at if they were biting the novel object. So we just put a toy inside the aquarium and we see if they were biting the toy or they were staying away, or how long are they close. So there are different metrics that they are trying to be standardized across, not only across species, but also across different taxa. So to be able to compare these.
After that, you have to somehow cluster those different experiments into different behaviors, like explorers.
Yeah, those are very defined categories to be able to keep the field kind of like similar, and then you can make some comparison between species.
What's the size of your samples, more or less, just so
for the individuals that were in the field, so if we have like wild individuals, we have behavior for around 300 individuals, and we did the genetic analysis on almost 200 of them. So, and for the ones in the laboratory, but I don't have the data yet. So the experiment, the results I show here are from the field. So it's around between 150 to 200 individuals. And for the experiment in the lab, we only did personality tests for 60 individuals. So that's going to be a smaller sample size, but we don't have the results.
And how long does it take to get this distribution you have? I mean, you've been harvesting for fishing for a year?
Well, for the first experiment I did, the experiment lasted 3 months. I think the one in the field, and the one in the lab, I don't know. Marco did it. So I wasn't here. So it was just a few weeks, I think. But it was a long, like it was so the behavioral taking the information, it was kind of just, you have to just put the tags on the fishes and wait for 3 months and then you collect the data. But preparing all of this, it was way longer. So and then after that, getting the results out of this, so the behavioral scores, that's what I need. I just need a number for each fish representing the behavior. This is what takes longer because you have to analyze the data. I think is the hardest. I don't know, and I shouldn't even talk about this because
No more questions?
I have another question,
Yes.
and is when this relationship between the genes and the behavior. I mean, it is known what protein does this gene produce. I mean, there is a mechanism that goes from the gene to why the behavior might be different.
Well, it depends, of course, in the specific gene. For example, for the clock gene, it's very well studied, all of the different components of it's called the clock mechanism of the circadian rhythm. So it's all of the molecular underlying, all of these, and there are like maybe 20 or 30 genes related and how they interact with each other. So there are some that it's more known, other that it's not as much known, but for them, for the clock gene, they know the exact same process that each component adds to this other one and then this one to the other one. So there are some, there are some that we don't know, some we don't know that much.
Okay. That's all. Now we remember that we have some weird answers. Thank you very much, Marta.